So we'll go this way. So you will know what I'm about to say because you'll see the slides that are coming. So what is my general approach? Yeah, unfortunately, then. Sorry? Microphone. Yes. Well, I didn't have time to say anything meaningful because. So fortunately, I don't have very important animations, and you will see my slides in final form, which is fine. I will guide you through them. So basically, my general approach here is that there are some basic principles we all do adhere and understand and agree on when we study this transition from chemistry to life, and that is that you need 4 more or less consecutive steps to occur. You have to have the necessary feedstocks, and these are the elements which we have to have available on the locality, the planet where they have to be also accessible, not just available. You have to then somehow have a chemistry that builds the molecular building blocks in a persistent and very robust way. We actually, for life as we know it, we know there things like lipids, amino acids, nucleotides. And then they have to be in an environment in which they can form polymers. And not only that, but those polymers have to be functional, not just tar, as my colleague Steve Benner likes to point out, happens quite often, maybe on planets like LS34, forgot the whole number now, used to know them. And then, of course, you get to this protocell step, the one that I told you we as a community are kind of shooting for right now. So the important thing about this to understand, and Steve and I have been pushing this for many years to remind people that from the chemistry that produces the molecular building blocks to the protocell, there is a supply chain that has to be robust and persistent for a long period of time. Why? Because these protocells do not yet have the enzymes, the machinery, which Georg talked about, like Rubisco, to actually produce those building blocks. So the environment in which those building blocks were initially synthesized has to actually persist, and persist over a long period of time. How long, we don't know, but it has to be long enough that these protocells have time to explore genetic space, to explore actually the opportunities that are afforded by Darwinian evolution, to the point that it can become sustainable and does not need the supply chain. It's kind of, think of it as the initial protocells as babies, and they need baby food, and if you only give them adult food, like carbon dioxide, they die. So there is no Rubisco. So not only is this supply chain important to persist, but also it has to persist in the same environment. So the environment that produced the molecular building blocks has to be also the same environment in which actually the protocells are evolving. So you cannot do it in 2 different places and times. So this sounds like trivial, but it's actually, I do want to emphasize it as an important constraint here in building those models, in those geochemical models from that time. And so, by the way, there are other constraints which we always talk about, and many of the practitioners in this agree with. For example, one of them is those feedstocks, and the ability to build the molecular building blocks require concentration of some feedstocks which are not that easily accessible under the normal conditions we imagine for the early Earth. And we have to have environments in which we can concentrate them. So, for example, we need concentration of phosphate and borate, we need concentration of sulfidic anions and fixed nitrogen products, like here in this example of one of the successful chemistries for prebiotic building blocks by the Thomas Carell group. you can see that this can be accomplished in constrained aquifers, in lakes where you have constraints, so that under the changing climate conditions, you can have wet-dry cycles, and that allows for concentration. Usually, this kind of lakes are also not just evaporative lakes, but they have large catchment basin because they're closed basin lakes. And so that allows for a large area which leaches things like phosphate and borate and concentrate them into a smaller area where actually the lake happens to be. So it's not just the wet-dry cycling that we have here. There is also the other thing is we know that the early atmosphere of the Earth, by all evidence we have so far, was mostly oxidized. So it actually was lacking some of the atmospheric component which will allow you to synthesize reducing precursors. And these reduced precursors have to come from somewhere. So then the question is, do you have constraints from the point of view of how long these unproductive atmospheres actually exist, the neutral or redox neutral atmospheres? Do you need to invoke transitory reduced atmospheres, which actually should have happened because there were impacts, large impacts in the history of early Earth? So that's another thing to consider here. We put those into piecemeal models, but basically, to a great extent, we depend also on what our colleagues in geochemistry have done over the years. And this is a textbook example of how any rocky planet, similar to the Earth, would evolve very quickly on the timescale of less than 1 million years from having an interior which is fully mixed and hence producing reduced atmospheres to one which is differentiated. And that happens even to very small planets with the iron core. And then the atmospheres are pushed to be a lot less reduced, mildly reduced, and actually neutral. And the reason I'm showing you this textbook diagram is because Laura Schaefer, when she was a student and still working on this, actually looked at the physics of that, the geophysics of that, and showed that actually that same argument would extend all the way to what we call super-Earths, or larger versions of the Earth, which are up to 5 Earth masses in bulk mass. Now, this is important because when you go to the planets that are accessible to Laura, and Laura talked about planets which are bigger than the Earth, 2 times in radius larger and hence more massive as well, you should expect to see some of the same physics up to a point. And so this is an open question, but we do need to know the answer. If you look at this textbook example and really dig into what is actually, where is this information coming from, it turns out that very little of it is actually direct evidence, either from geology, we obviously don't have, or for studies in the solar system, direct studies. It's mostly indirect and mostly theory. So my biased opinion here is that we still have a question mark on whether this is a common occurrence. This development into neutral or redox neutral atmospheres is a common thing and happens at the timescales we imply here. And the only way to really solve that problem with the current availability of the data that we could get from the solar system is actually go beyond the solar system and rely on a large gallery of atmospheric studies like the one that Laura is actually doing. So all of these things have to be taken not only with a grain of salt, but with the understanding that there is theoretical models and there is evidence that we can use or not use. And so basically, for most of the 3 or 4 successful chemical pathways for prebiotic chemistry, you do end up with some basic constraints, like you do require liquid water with closed basin aquifers. And then the question becomes, again, how do you get those reduced precursors that are needed for the chemistry? You do have some very small amounts of things like hydrogen cyanide and carbon monoxide in the atmospheres of those planets. But they are such small amounts that they're not possible, especially as stoichiometric components, to drive that chemistry. So basically, one solution is what you saw from Steve Benner's group work. You rely on transitory periods of time in which you have large impacts in the early Earth, which produce those reduced atmospheres for a period of 1,000,000 years or so, and you do all of that chemistry during that time. And we'll hear from Steve this afternoon, I'm sure, more about this, the particulars of that geochemical model. I'm certainly agree that this was happening on the early Earth and that it is a very good way to produce those precursors. My personal preference is that we not just produce them and then do everything while they're being produced at the same time, but find ways to store them as sedimentary and other deposits which are layered on the bottoms of that lakes. And then you think in terms of a whole sequence of events where the preparation of the conditions for the emergence of life takes a long period of time. You can wait for several million years until this kind of lakes are and sediments and deposits are available. They go through dry wet cycles and evaporation, and then you use them later on when the time comes. To me, that is, from a planetary perspective, it's a little bit more robust and easy to imagine. And I want to leave you with a kind of a visual perspective of what this looks like by, again, the best ideas that people have in this field. The early Earth is both similar and different from today's Earth or more recent history of the Earth, mostly in the fact that continents were smaller or even completely submerged. There was not much in terms of continental subaerial land. In any case, it was less than what you would consider a continent like Australia today. That's plenty for our needs in prebiotic chemistry, but it's just good to know that you have a lot of liquid water sloshing around and mostly big islands. There is nothing bad about that, big islands. There's a lot of evaporative lakes that occur on this kind of islands. Just look at, well, this is the problem with not having the actual presentation. Look at Iceland, which you can't see, overlaid by a map of Mars, Gale crater. But Iceland is a good example of something like would have existed. I know geophysically it's a different structure. The big island of Hawaii is a better one. But at the time they were bigger, and that's why I like Iceland as an example here. And we know that this kind of evaporative lakes existed on Mars. In fact, they are better preserved there than on the Earth. Mars preserves its ancient record in superb detail compared to the Earth. And to some extent, a lot of my own inspiration has come from working with people who actually have discovered these structures on Mars and trying to imagine or model how these environments actually occurred here on the Earth. And as I promised, I wanted to give you one example of how these constraints can be approached. So basically what I'm thinking about here is that you cannot just take constraints, how would I say, post hoc, and just say, okay, my chemistry works because somebody said that this kind of geochemical constraint is available. You have to actually constantly build those models to be self-consistent. The model has to be complete and self-consistent. You cannot just have piecemeal, this works, that works, but if you put them together, there is no such self-consistent solution. And the question is, what do we do? Well, we constantly introduce new constraints. And again, it's an iterative process. And I've been thinking a lot about that. You know, what other constraints can I steal from my colleagues who are the chemists? You know, they come up with new little solutions for parts of the network. And one which they have generally emphasized but failed to solve is the requirement for homochirality. We all know, since Louis Pasteur, you see the tartrate crystals on the old wine cork here in the picture. That's how he determined 180 years ago now that life is unusual in being homochiral. It was dissymmetric, as he would call it at the time. And basically, this is a requirement which is important. We know that. We understand why it is important, why it's essential to life that the polymers are homochiral. We also know very clearly that homochiral is the right word, left and right. But while some molecules, the sugars and the nucleotides, have to be of the right variety, the corresponding proteins made of amino acids have to be of the left variety, and so on. So we call this a homochiral system, and we understand why it doesn't work otherwise. I was particularly kind of pushed in that direction myself because I was worrying about something which in synthetic chemistry is called the arithmetic demon problem. If you have a synthetic network which consists of multiple steps, which is in fact true for all the prebiotic chemical networks, then your yield at each step accumulates to a very problematic final yield if you don't do anything in between, like purify or separate and so on. You lose yield, even if you have fairly good yield in a multi-step system. And so one sure way to lose yield and to end up with very little or no product is racemic solutions, because you start with non-chiral simple molecules, and they, synthesize bigger and bigger product. At some point, you suddenly have chiral ones very soon, and now you've lost half of your yield for productive, and then they each have stereoisomers, and you know what happens next. So the homochirality problem had to be solved early, very early, during that prebiotic network time. And so as a physicist, background in physics, I really cared about what is exactly that the components that you have to have in order to get the homochiral system. And there are three of them. You, of course, need a chiral agent. You need some way to beat the chemistry, which is constantly pushing 50-50 racemic solution, left and right, to a little bit of an enantiomeric excess. EE there stands for enantiomeric excess. Right enantiomer, left enantiomer. The right is a little bit more than the left. That's an excess of 2 to 5%. Then you have to amplify it. 2 to 5% is only good for a very, very short time because chemistry immediately balances you out. So you have to amplify it quickly to 100% and then keep it that way. And keeping it that way means that it has to propagate through the network. You cannot do it for one of your building blocks and then assume that somehow miraculously the rest of them will follow suit. It doesn't work that way, as I discovered. And so this last third component, which is the network propagation, was very important to us. And when we thought about it last year, we thought of it as propagation of information. And so we call it in homage to Francis Crick's central dogma of biology. We call it the central dogma of biological homochirality because you need to solve that propagation of chiral information problem. So where do you find the chiral agents? Well, you find the chiral agents mostly in physics. So a lot of my colleagues in physics have actually and still working on chiral agents themselves. I've tried many of those, apart from 7 and 8, which require chemistry skills, which I lack. But Donna Blackman was always a very good teacher to me, and she showed me how some of this asymmetric chemistry works. Unfortunately, none of them is very useful for the third part, which is the network propagating aspect of it. It can provide amplification if you have the right chiral agent, but then how do you propagate through the network? So, long story short, through all kinds of interesting tribulations, I found out about this newly discovered at the time, several years ago, effects in physics or physical chemistry, which is called the chiral-induced spin selectivity effect. Ron Naumann and David Waldeck had discovered it. It had to do with the spin of the electrons. And really, the way it works is you take a chiral molecule. They used chiral peptides, alpha helixes, or DNA itself, a double helix, and they would push electrons through it, and they will see that it acts like a spin filter. It actually filters one of the spin, and then they change the chirality of the molecules, and it reverses the spin effect. So the effect is extremely interesting to me as a physicist. Why? Because it's inherently a quantum effect. You know, the electronic spin and what's happening here has some similarities to the Pauli exclusion principle. But it works at room temperature in water as a solvent. That is what we need in the emergence of life chemistry. So why not try to figure this out? Well, it took us 6 years to get to. We applied it in reverse. So in other words, if you think of it in terms of, again, as a physicist and an operator, it's an abelian, you know. If they can push electrons and filter them through a chiral molecule, can I use already biased electrons to synthesize a molecule in the right chirality, meaning right-handed or left-handed, depending on whether the electrons this way or that way. And that's what you see here. The idea was, if I can crystallize that compound, maybe I can separate the two enantiomers. Because when crystallization happens in room temperature in liquid water or solvent, and you approach a surface of some sort, not only is the electron spin potentially biased, but more importantly, the molecule itself will have to get charge polarized. The delta plus and delta minus in these two examples is actually the expected charge polarization when you approach the surface. It doesn't matter how you approach it, you tumble, you will have that kind of a polarization. And that polarization, because of the chirally induced spin selectivity effect, will also transiently be accompanied by a spin polarization. It has to be. We calculated it, and we expected it to be a big effect. But the crystal that forms has to be a special type of crystal, and that was actually the trick. It had to be one of those conglomerate crystals where the left-handed and right-handed will separate from each other. And so then the question was, okay, so they're rare, but are there any examples? Okay. Are there any examples? Yes, there are a few examples. And by Occam's laser, it should be in one of those groups of subsystems in the metabolites, the L-peptides, or the sugars. And so we started with a few of them. Historically, tartrate, tartaric acid, is the one that you would like, because that's what Pasteur did, and metabolism will solve the problem because you need metabolism for everything, so if you start there. So tartrate, there is a prebiotic way to make it. There is a prebiotic way to turn it into malate, so immediately part of the Krebs cycle, but you immediately have a problem. Either way, whether you go reverse or forward, you end up with a non-chiral molecule. That was a dead end. So maybe it's the amino acids. People always, in the past 50 years at least, have thought somehow that homochirality will start with the amino acids, even because of the meteoritic discoveries and so on. So yeah, there is one amino acid among the canonical ones, asparagine, that forms perfect conglomerate crystals. There are two more, but they're special salts. Again, because of its solvation properties, it didn't work. And so the final one, and literally there is no other one left, is among the sugars, and it happens to be a precursor of RNA and DNA. And again, from a paper by Thomas Kirell's group here, you can see that it actually is a molecule, this aminoxazoline, which combines different chemical pathways. It's the one which is called REO for short. And of course, REO, as it should be, is not only left and right, but it actually comes in a family of others. And it turns out that if you put it near a spin-biased surface. Magnetite is perfect for that. It separates like a charm. It really separates really nicely. And these experiments, we are now doing them multiple times, really produce homochiral results in literally 2 steps. So you have a good, very strong chiral agent and an excellent amplification. And because of what I just told you, it also happens to solve the third problem, the possibility to get through that information, chiral information through the network. And that's to a great extent thanks to actually work which has happened literally in the last year, some of it in Carell's group, some in Berkeley and other places, which was anticipated going back to the 25 years ago by Schimmel and others. Then in the Sutherland group, they had actually done some of that, although they didn't think of it in those terms, but it worked, stereoselective aminoacylation. So the central dogma may actually work. You may start with a single enantiomer component, which then propagates all the way to the Krebs cycle and to the biologically homochiral. So how would that actually work in terms of early geochemical problem? Well, you have to have lakes which accumulate this stuff, the magnetite. We see them on Mars. There it is. There is a very good example. We don't see it on the Earth yet, but it turned out that people hadn't actually looked for it that much. So for the first time, we're actually going to early Archean lakes like Tumbiana and collecting samples, and I was there too. And we are able now to reconstruct this surface environment in some form and fashion. But it's only one constraint of many. So this is a work in progress, and it will take all the different pathways that people are developing by the different groups and many more constraints to actually piece together all of this and be able to one day say, well, we have a good general understanding of how this works. And maybe that will help us, at least that's the goal, help us build this elusive protocell in the lab. And then the really exciting work will start. Thank you.